Okay, great. Oh, I think we have, oh no, seems like the echo's off. Yeah, yeah, exactly. Yeah, so I think you have to have your sound off. Okay. Well, thanks, Lois. That took us a little while to get going there, but sorry. So, what I want to talk about today is just, a little bit about what we have at the Academy. And um, I've been at the Academy of Natural Sciences since 2014. I moved here from the Field Museum in Chicago, which is also one of the largest collections of bird specimens, houses one of the largest collections of bird specimens in the world. That collection is about 500,000 or 600,000 specimens. To give you a sense, our collection is about 225,000 specimens, something like that. We don't actually know the number exactly because although it was the first collection of its kind database, when it was databased, not every specimen was entered in the database. We don't know why. So we're in the process of rehandling every specimen in the collection to verify the data, enrich the data, because back when they first databased the collection, they only, indicated things like location, date, and species. They didn't indicate all the other information that's on the specimen in that database. We do that now. So all the new material has that data captured, but we're going back and doing that with everything. So we're sort of enriching that data, and that's all served publicly online. You can find it if you go to GBIF, which is a system online that sort of conglomerates collections from all over the world into one portal. Anyways, when I got to the Academy in 2014, I told myself that every day I was going to open up one case and peruse the specimens in that case and just sort of get a sense for what's in the collection. And I've managed to do that a lot. If I'm reading a scientific paper on something and I'm curious what we have, or I'm curious about something in that paper, I can go to our collection. It's like a library, right? I can just walk over to the cabinets, open the drawers, and look at the things that we have in those drawers. But the reality is I'm so busy that I don't get as much time as I'd like to do that. And whenever I do get time to do that, I'm always amazed. I mean, even to this day, 10 years later, I find interesting things that I didn't know that we had in the collection. And it's a fun thing to be able to explore a collection like the Academy's collection. And I'm sure all of the other inhabitants of the Academy historically had the same kinds of feelings that, they went around that collection and found interesting things that others had collected over time. And this little kind of mural on the slide here is just a few of the individuals that built the collection, not early on, but sort of in the early 1900s, late 1800s. The person on the left there, or on the right, on the slide right, is Mel Curator. He's holding a curacao and a tinamou that he collected probably in Colombia, although he collected specimens for our collection in Peru, Bolivia, and Colombia. And then the two gentlemen at the bottom in the middle are Meyer de Chauncey, who's famous for doing work in China and also South America. He wrote the big field guides to those at the time. Although the Birds of South America, the original field guide that he wrote, was for folks like us, in comparison to what we're used to, almost unusable. And the first time I went to South America, I carried his field guide because the birds of Ecuador didn't exist. There was no field guide to the birds of Ecuador the first time that I went there. So we carried the birds of Colombia, we carried Meyer de Chauncey's Birds of South America, and then we carried this other big book called Birds of the High Andes. So you had to have a little library with you. And now you can just, have your cell phone. And a lot of times you can have a field guide on your cell phone, or you can have Merlin on your cell phone to look things up. It's amazing the tools that we have now. And we actually have a lot to thank for that. A lot of that is folks like this. Information that we have gathered over the years has led to these great field guides that we have. And this is information that comes from museum specimens in large part. It's a combination of like museum specimens and a lot of community science. So a lot of birders out there gathering information and pictures and that together with museum specimens really sort of has matured ornithological science in a way that we as scientists and birders have benefited from that. The other person in this picture next to Meyer de Chauncey is James Bond. And I think probably lots of you have heard this whole story about James Bond. He was an ornithologist. He was our ornithologist at the Academy. And basically Ian Fleming stole his name. That's the short story. Sometime I can tell you the longer story, but there's a whole book all about it. Basically his name was kind of a manly name. Ian Fleming. This is the name I'm going to take.
To change it.
And sure enough, it was our ornithologist. So I mentioned that our collection houses about 225,000 specimens. This is the second largest university-based collection in the world. We're second to Harvard, and we're about neck and neck with the University of Michigan. The University of Michigan has a large university-based collection as well, although they don't grow at the same pace as ours. Our collection is growing by a few thousand specimens a year, depending on whether we do field work. We do a lot of salvaging. That's how we grow the most. So we do things like we get building killed birds and prepare those as specimens. We go to other museums, like North Carolina State Museum always has a freezer full of things that they don't want or that they don't have time to prepare. And we hop in a car and drive down there and empty their freezers and throw them into a cooler or a few coolers and drive them back to the academy. And we prepare those specimens. We've done that with folks on the West Coast as well. So we've added albatross to our collection and Corey shearwaters and all kinds of things that we wouldn't get right here in Pennsylvania. And that's mostly how we grow. And our collection mostly consists of bird skins. We do have pickled specimens, which are alcohol, specimens stored in alcohol that have been fixed in formalin so they can be used for anatomical work. We have some nests and eggs, but not a lot of those. Those were traded away many years ago to the Western Foundation of Vertebrate Zoology. So our egg collection is about one large case worth of specimens, not the vast collection of eggs that it used to be, but our bird skin collection is pretty vast and pretty comprehensive. Like Lois mentioned, it's global in scope. You can see things from China all the way to the tip of South America in our collection. What I want to do is I'm going to touch on a couple of different things. I'm just going to basically walk you through some specimens. So I sort of developed this talk when COVID hit. And normally for the Delaware Valley Ornithological Club, I would give a talk on sort of the kinds of things that we were doing and the importance of collections. And then people would come back and do a behind the scenes tour. And it was a pretty popular event. I don't even remember how many people, 40 people sometimes that would come, or 50 people to come to that event. We'd have to break up into groups because it was too many people to have with one person at a time in the collection. And but when COVID hit, of course, we couldn't do that. So I said, well, why don't I, put together sort of this virtual thing? I can go to the collection by myself. I can take pictures, and then I can put together this kind of story about the iconic specimens in our collection. And of course, this is just a smattering of them. This is what it would be like if you walked into the collection and I took you on a tour and we just sort of bounced around and looked at fun cases in the collection. So we're going to see some endangered things. We're going to see some extinct things. And we're going to see some little known and just kind of interesting things. And a few of these are things that my lab is doing research on, and I'll tell you a little bit about those. But a lot of them are things that are just, they're sort of little interesting stories about them. So let's start out with extinct things that are in our collection. And yes. Unmute. Okay. My head is kind of angled, funky. People are probably seeing my chin. So is all good? Can folks hear me? Okay. So let's start talking about some of the extinct things in the collection. These are obviously sad stories that, these are extinct birds. And we just recently heard about slender-billed curlew. It may be in the news if you've heard about that. It's a shorebird that recently has been declared extinct, a Eurasian thing. Curlews, unfortunately, have a history of going extinct. There are other ones, and the one I'm going to tell you about now is an extinct one. But fortunately, we do have specimens of some of these things in the collections around the world. And sometimes that's all we know of these things. Sometimes we get lucky and these things are rediscovered. And sometimes it's specimens that can help us to do that. Oops, let's see. That can help us to, learn about these things so that we can rediscover them. In this case, this is a bird that is extinct. So this is an Eskimo curlew. And the specimen that's pictured here is a specimen that was prepared by James Bond. So he wrote his name on the tag. So the preparator of the specimen writes their name on the tag. And usually you get that tag will have a number. That's a catalog number that researcher wrote the data on in a catalog that is, kept at the museum along with the specimen. Now, he did not collect that specimen. This was a time when Eskimo curlews were actually on the decline. So the decline began in the 1870s. And by 1900, pretty much any sighting was noteworthy. And the story goes that a lot of the decline of these was due to unregulated hunting. Shorebirds are hasty. They were hunted, and in this case, market hunted. And in this case, what happened was James Bond left a freezer in Barbados. So James Bond was an expert on birds of the West Indies and spent time on lots of islands in the Atlantic and the Caribbean. And he basically left a freezer at a hunting club and said, hey, if you guys, shoot things, leave them in the freezer, I'll take them for the collection. Not expecting to get an Eskimo curlew from Barbados. These were birds that really traveled through North America. They were found in Texas on the Texas coast and went all the way to southern South America to Argentina. So this is a somewhat unusual record, and unfortunately was the last specimen record of this species ever known. And it was collected in 1963, so not all that long ago. And the last confirmed sighting in South America was 1939. So this gives you a sense for, this was a later sighting when this thing was really, almost non-existent. And the last reliable sighting was in the late 1980s. You never know. We could be lucky, but very unlikely at this point. Oops. This is a bird that we used to have here in Pennsylvania. This is a heath hen, and heath hens are greater prairie chickens. This is a subspecies of greater prairie chicken that was found along the Atlantic coast. And these, again, were one of these birds that was wiped out by market hunting. So, market hunting is unmanaged, just, hunting to sell huge quantities of meat, basically, right? We know that managed hunting can be good for populations in terms of science being done. Things like ducks. Ducks are one of the few groups of birds here in North America that are really doing well. And that's because they're well managed, and hunting is part of that. We have preserves that are funded by hunting. There's a reason to protect them. In this case, when it's market hunting, it's no holes barred, no science, no management, just crazy hunting. And that led to their extinction. These were originally found from Maine all the way to Virginia. And here in Pennsylvania, they were found in serpentine barrens. These are grassland birds. So their relatives in other parts of the world also live in grasslands, like the prairie chickens, the greater prairie chickens that you'd find in the Great Plains or in tall grass prairie. And then there's Atwater's prairie chicken in Texas, which is still in existence, but pretty endangered. It's heavily managed. They're working on trying to revitalize that population. And these were basically extirpated in 1932. And the last populations were on Martha's Vineyard. And we have several specimens of these. The interesting thing is people have used these specimens to sequence the genomes of these birds. So modern specimens that we collect tissue samples, and those tissue samples go in our frozen tissue collections where they're kept in good condition for genomic sequencing. So we can do all kinds of really great work on those tissue samples. It's a lot harder from specimens, but the technology's gotten so good that we can get whole genomes, even from old specimens from the 1800s. So some colleagues of ours borrowed toe pads. We scraped a piece of a toe pad off the bottom of the foot of these specimens, and they were able to sequence genomes from them to learn about their population history. This one is also probably familiar to you. This is Carolina parakeet, another one of these sad extinction events. This specimen happens to have been one that was collected by John James Audubon. We have about 300 bird specimens collected by Audubon in our collection. And this one even has an original tag. So if you look at the picture on the right, the lower tag on that specimen is the original tag with Audubon's handwriting on it. The other tag that says Academy of Natural Sciences on the top is not the original tag, but we put both on there to make sure that we keep the data with the specimen just in case that old tag may not be as archival as the new one. And this was collected at Fort Leavenworth on May 4th in 1843. The last population of these was in North Carolina or South Carolina in the 1930s. So pretty far back. Yes. Yeah, so this is called a study skin. And these study skins are a special kind of taxidermy for scientific work. So if you were to do taxidermy for exhibit, you put wires inside the specimen and you create a body that poses the animal, in some sort of natural pose. But the taxidermy that we do, they're called round skins or study skins, is done in a way to store specimens in drawers. So these are stored lined up in drawers so that we can basically optimally fit as many in a space as possible. And for some things, we're running out of space. We only have so much room in the museum. For songbirds, we have lots of space, but for snowy owls, we have so many snowy owls, we still take them, but we have to, buy new cases, fit new cases in the little corner somewhere to store these specimens. And it's actually important to continue collecting specimens like this because the way that these specimens work is every specimen is a unique individual, right? So it's just like in this room, we're all unique individuals. We all look different. We all have different measurements. We all tell a little bit different story about biology and about life. So those different individuals are valuable. And if we want to study things like change over time, we need series of specimens over time. So if we stop taking on new specimens, we'll suddenly lose that ability to tell those stories and ask those questions about how things have changed over time. And there are all kinds of really interesting stories that have come out of things like window strike birds. So colleagues of mine in Chicago were collecting window strike birds off of one building, McCormick Place, which is a famous building on the lakeshore of Chicago that has killed more than 40,000 birds from window strikes. It's now finally has the windows treated because last year there was a huge, huge migration event, probably one of the biggest migration events that any birders that I know of have experienced in Chicago in modern times. And that migration event, not all the window shades were closed, some of the lights were on, and at dawn, when the birds ended up over Lake Michigan due to the northwest winds, or sorry, the southwest winds, because it was a spring migration event. Those birds collided in mass on the building. So there were any windows that had lights on and the shades open. There were birds all over the ground in front of them. The ones that had the shades closed, there were not birds in front of those windows. And my colleagues have been studying this for decades. So they've been able to learn about what are the factors that lead to these deaths, but they also have been able to use the specimens that they've retained from all of these kills. And those specimens tell all kinds of other interesting stories. So they've made good use of them. One thing we know is that body size is changing in a whole bunch of different songbird species. And we don't know why that is. It's probably climate driven, but basically wings are getting shorter. Bodies are getting smaller. So there is a change. This is what we call microevolutionary change. Could be human caused through climate change, could be something else. We don't know for sure, but these are patterns that we can only find with big series of specimens, and they had those. So it's why we retain, why we continue, picking up specimens of common things when we can. So yeah, that's a long story for the answer to the what is this, but it sort of gives you a sense of what these things can be used for. It's all kinds of different interesting things. These are the Cuban ivory-billed woodpeckers, a male and a female. And these are actually what are called type specimens. That's why they have these red tags on them. And type specimens are the specimens used to describe a new species. So when a scientist goes out and we discover something new, we usually write a paper in the scientific literature and we designate a specimen as the type. And what that means is that specimen kind of becomes the icon for that species for any future studies. So when people want to, if someone found another population of ivory-billed woodpecker somewhere and they wanted to find out if it was different than these, they would compare their specimens with these type specimens. And what's interesting is, we used to have ivory-billed woodpeckers all over the southeastern United States, and they were in Cuba. And there was this big question, well, what if the birds are still in Cuba, but they're extinct in the United States? Could we bring some into captivity? Could we raise them? Could we then release them back into North America and then have this ivory-billed woodpecker back here in North America? That's a good question. It's kind of what we did with peregrine falcons, right? We extirpated peregrine falcons from Eastern North America, and then we took birds that had been in captivity and we released them all over cities in Eastern North America. Now they're pretty common. I see them out my office window at the Academy of Natural Sciences, and I saw them in Chicago at the Field Museum outside my window. Well, that's not always a good idea. You really want to study the birds and know something about them beforehand. You want to know whether the things that you're releasing back into that environment are the same things as what you extirpated. So in the case of these ivory-billed woodpeckers, before we really knew what was going on, colleagues of mine decided they were going to sequence DNA from old ivory-billed woodpecker specimens from North America and from some of these birds from Cuba. And what they found is that the Cuban birds were genetically pretty different from the North American birds. So what that means is if the birds were still in Cuba, which we don't think they are anymore, we don't think any ivory-billed woodpeckers are extant. There's no good evidence of that at this point. If they were still there, then you probably would not want to just sort of take them and bring them back into North America. It's really a different beast, a different creature. So the specimens can tell us lots of interesting stories. And the last credible records of ivory bills in the US were in 1944. In Cuba, it was in the 1990s, the last, or 1980s and 1990s. This is kind of one of my favorites. And this one is one of my favorites because it's such an unusual specimen. This is called Stephen's Island wren, and someone mentioned that one of the club members was in New Zealand, probably on George Armistead's trip, I'm assuming. Yeah, because George is posting all kinds of. George is a good friend of mine, and we were actually just in Brazil together this fall, and I know he's on a trip to New Zealand. Well, this is from an island off the coast of New Zealand. So I think George's trip is going to islands right now, and they're going offshore then to go look at seabirds by boat. Well, this specimen came from Stephen's Island, an island off of New Zealand, and there was a lighthouse keeper named David Lyle. And in 1894, it was named after David Lyle, who basically discovered this thing. And he discovered it because his cat or cats, it's a little bit debatable. We don't know for sure whether it was a single cat or a colony of cats. The story is that it was Tibbles the cat. There's actually a name for the cat. And the story is that Tibbles the cat was bringing in these birds. And Lyle was interested in natural history and knew how to prepare skins. Not very beautiful skins, but they were skins. And he prepared these skins and then sent them back to museums. And this skin originally comes from the Baron Rothschild's collection, which is in Tring in the UK. And that collection was at one point split up. Half of it is still in Tring and half of it is at the American Museum of Natural History. And at some point in time, our curators, probably Meyer de Chauncey and James Bond, traded this specimen for something. I'm not sure what they traded it for, but basically we got this specimen probably from the American Museum. And it's one of the types from the series of specimens that were used to describe this species of wren. And it's not a wren like our wren. It's a group of things called New Zealand wrens. And it's thought that this bird was flightless. So a flightless songbird, not something that you see or hear about very much. And of course, that made it much more prone to cats, right? We know that even flighted birds are prone to cats. Cats kill 3 billion birds. We know that windows kill 1 billion birds a year in the United States. Feral and outdoor cats kill 3 billion. So if you have friends that have outdoor cats, something that you got to really get people to stop doing. It's a really bad thing for the things that we love, birds. This extinct bird is not a whole bird. This is one of our eggs. And to give you a sense of the size, I'm not sure if you can see on the screen, but this egg is about this big. So a huge egg. This is an elephant bird egg. And that's a drawing of an elephant bird egg. I don't have a picture because no one has a picture of an elephant bird. They were extinct a long time ago. Basically, they went extinct in 1000 AD. And these things were about 1,200 pounds. So a huge bird, 9.8 feet tall. And so that's 3 meters tall. And they were from Madagascar. So these are huge ostrich-like, they're ostrich relatives. And we have 2 eggs in our collection, and it's pictured here next to an ostrich egg, which you already know is pretty big, right? So it dwarfs the ostrich. It makes the ostrich egg look teeny. And one of our eggs was fragmented and is kind of glued back together. This one is not fragmented, and some of these eggs have been CT scanned and actually have embryos inside of them. Ours does not have an embryo. So this is kind of a subfossil egg of this huge, crazy bird.
Question, how did you get that?
Back in the day, people like Meyer de Chauncey and James Bond and even people before them were really good about looking for things. These things were kind of in the they were out there in the natural history trade world. So they purchased them or traded them with other museums. Yeah. So I don't know the story behind those specific things, but they're pretty hard to get now. Although what's interesting is if you go to Madagascar, you can actually find bone fragment or the shell fragments of those eggs. And there was a paper study recently that sequenced genomes from shell fragments that were just, found in middens and things in Madagascar. So, but a whole egg is a lot harder to find, like that one. I don't know exactly, but at least 12, at least it dates back to at least 1000 AD. But probably before that, I don't know the answer. Yeah. I don't know if anyone's ever dated our egg. Yeah. So good question. This is a really, really strange specimen. This one is called Bogota sun angel. It's a hummingbird that was bought in one of these kind of markets in Bogota where they sold animals and animal parts and things many years ago. It was purchased in 1909. And colleagues of mine were debating, is this a hybrid? Is it a real thing? No one knew this bird in life. And in the neotropics, I like to call South America the bird continent. It's the most diverse place bird-wise anywhere in the world. If you want to see a lot of birds, the best place to go is South America. You can go anywhere and just see hundreds of species, if not more, if you spend enough time. And so every so often, people rediscover things that were thought to be extinct. So there was a real big debate about this. Is it real? Is it just out in some little inter-Andean valley that's hidden away that no one's explored? And colleagues of mine in 2009 decided they were going to sequence DNA from this thing. And we loaned them the specimen. They cut a little toe pad off of it. And Jeremy Kirkman, who is actually a colleague of mine in graduate school at Louisiana State University, he and Chris Witt, also a friend of mine who grew up here in Philadelphia, by the way, and spent time at the academy as a kid, and then some other colleagues sequenced DNA from this thing. And basically, the DNA that they sequenced was mitochondrial DNA. This is DNA passed on from mothers to their offspring. And that mitochondrial DNA was different enough that it told them that this thing was a valid different species that was probably extinct, but who knows? Maybe it's still out there somewhere. Now, the funny thing is, in 2018, a study examined additional data. And that included data from a hummingbird that was found in Colombia that people originally thought, basically they thought they had rediscovered this Bogota sun angel. And it turned out that when they sequenced DNA from that thing, they were able to sequence a lot more DNA. It was a lot easier to sequence, nuclear genes and mitochondrial genes. And basically that bird and the Bogota sun angel had the same mitochondrial DNA, the same matrilinear DNA, but then they had a mix of DNA in their nuclear DNA that suggested that they were both hybrids. So it turns out that this is probably a hybrid and not a distinct species. So kind of a weird, interesting specimen. Okay, so that's where if you have specimens in collections, you can go back to them and study them and learn new things. Sometimes the story can change. So let's talk a little bit about endangered species in the collection. Have any of you been to Guam? So my parents actually lived on Guam. Right before I was born, my dad was in the Navy and they lived on Guam. So my dad always likes to tell me that he's seen a Guam rail. I don't believe him. He's not a birder, so I don't think he ever saw a Guam rail, but who knows? Back when they lived there, there were still Guam rails running around on the island. And these birds are critically endangered. They're extirpated from Guam because of the introduction of a snake. The brown tree snake was introduced from Australia and has wreaked havoc on native birds. Island birds are really sensitive to introduced species, whether those are cats or snakes. And basically by 1987, these things were, there were 100 birds or less than 100 birds in 1983. By 1987, they were extinct in the wild. And in 1983, they started a captive breeding program, and this was set up in zoos. The Philly Zoo is one of those zoos that's helped support that program. And now they've actually brought Guam rails back, but not to Guam. to an island off of Guam called Rota. And Rota, they were able to extirpate all land predators, rats, mice, and because of that, the Guam rails can survive on the island there. Let's see. Oh yeah, and also Cocos Island is another island they've done that. So the captive breeding in this case has been successful, and these are actually modern specimens from that captive breeding program. Of course, sometimes in that kind of situation, birds die, and fortunately, we have a pretty good relationship with the zoo, and they will give us material like this that's kind of irreplaceable. You know, now we have tissues in the collection that people can use to study the genetics of these things, and you know, there are some some modern specimens in this case. This is a Zapata rail, and I'm going to show you kind of a couple of different things that James Bond discovered and studied. So this is a picture of James Bond in the upper right. And James Bond is well known for having found the only nest and eggs of this species, which is from the Zapata swamp in west central Cuba. This is critically endangered. There are less than 250 species of this, individuals of this thing left. It's virtually flightless. And one of the things that we think is happening is introduced predators. There are lots of cats on Caribbean islands that cause problems for island birds. And also burning of the swamp is causing reductions to their population. And this one is kind of a weird one that's had a lot of ups and downs. This is the Bahama nuthatch, which on some taxonomic lists, on some checklists, you'll see it as a subspecies of brown-headed nuthatch, which you can head over to Delaware and see brown-headed nuthatches if you want. But there is some data showing that this is actually a distinct species. And sadly, it's sort of been up and down. We thought it was extinct. We thought it was extirpated from the island, and which means it's extinct if it's a distinct species. This specimen and one other were collected in 1931, and those specimens were used as the types to describe this distinct subspecies. But in 2019, Hurricane Dorian likely rendered the last birds extinct. There were a few birds that were refound by researchers, and I think since then no one has seen them again. So, you obviously get lots of strong hurricanes and, issues like habitat destruction, introduced cats. All that together is bad news for island birds. This is a success story, and this is a success story that has a little story about some of the research that we're doing. This is the California condor. How many of you have seen California condor? Yeah, so a decent number of people. I actually have not seen California condor. I'm looking forward to getting to Southern California one of these days and seeing some. Because when I started birding, they didn't exist in the wild, right? And basically in the 1980s, these things were extinct in the wild. And that's because they were down to, somewhere between 18 and 20 individuals. And U.S. Fish and Wildlife and a number of zoos worked together with the Peregrine Fund. They pulled all those birds out of the wild and brought them into a captive breeding situation. And this is, it's sort of a famous situation where they used puppets to feed the babies so they didn't imprint on people. And it turns out it was a very successful program, and they were able to release birds back in the wild, and now they're reproducing. They're in Utah, Arizona, California, Baja California. So, and they're expanding in California. So they're doing fairly well. Part of that is California requires lead-free bullets and shot in hunting. And this is one of the things that was wiping out birds like this. They were eating carcasses of animals that got away from hunters. And if they were riddled with lead, they were eating the lead and dying. So stop that and the birds can do okay. The story, though, that I want to tell you is actually about parasites on these things. So my lab studies a lot of bird parasites. We study parasitic lice and malarial parasites in birds. And it turns out the California condor had interesting lice on it. It had two species that were known and described way back in time. One of them described a species. One of them just, we know that it's on there, but it doesn't have a name. We're actually in the process of describing that new thing right now. But the one that had a name is this one on the slide here, and it's called Culpocephalum californici. It doesn't have a common name. Most parasites don't have common names, and this Culpocephalum californici is found on no other bird, only found on California condor, extremely specific to it. And it's sort of an icon for this thing that we call co-extinction. When you have a bird that goes extinct or a mammal that goes extinct, it may have parasites or symbionts that live on it or in it that are also unique. And we often don't think of that. As birders, we don't look at a bird and go, boy, I wonder how many things live on that piece of habitat. But that every bird is a piece of habitat for something, just like we're habitat for things, right? We have parasites and symbionts. We have bacteria that live in our guts and mites that live in the skin on our faces and lice that live in our hair potentially. And those are all specific to humans. So when we think about extinction, we should think about the extinction of these parasites. Well, what's interesting is when all these birds came into captivity, the veterinarians thought, oh my God, these things have lice on them. We have to kill them. So they treated all the birds with pyrethrum, which is flea powder, and they killed their lice. Now, fortunately, for a parasitologist anyways, fortunately, condors are big, and it's probably hard to treat a condor really thoroughly. But the story in the scientific literature was that this louse was rendered extinct when all the birds were brought into captivity, and no one had seen one. And then a few years ago, I get an email from the San Diego Zoo, and basically some of the zookeepers were handling a bird they were about to release, and they saw some parasites crawling on its feathers, and they grabbed them and threw them in a vial of ethanol. And they emailed me and they said, hey, Jason, can you tell us what these things are? And I said, sure, as long as they're not mites. I'm not an expert on mites. I would have to send those to somebody else. So I said, how many legs do they have? And they said, they have 6 legs. So I said, okay, that's good. They're an insect. That means I can probably tell you what they are. So I said, send them my way. And sure enough, when I looked under the microscope, they were this thing, Colpocephalum californiensis. So it went from being extinct to actually being fairly common. So now we have various zoos, Santa Barbara Zoo, the Peregrine Fund, they've all sent us samples from their birds, and they have these Colpocephalum californiensis, so they didn't go extinct. So we're working on a little publication to sort of say, hey, this thing really is still out there. So fortunately, it did not go co-extinct with its host when they went extinct in the wild. And I should mention one other thing. There is a reason that it's useful to have these parasites too. So when we want to study the population history of organisms like birds to understand something about what's happened over time to populations, we actually use DNA to do that these days. And that's because DNA accrues differences over time, and we can compare those differences among individuals, and we can use those differences to tell us what size the population was, whether that population has gone through what we call population bottlenecks. When populations get very small, it leaves a signature on a group of birds' DNA. And we use techniques called population genetics to study these kinds of things. Well, bird DNA evolves at a certain rate. But what's cool about parasite DNA is it evolves at a much faster rate than that bird DNA does. And what that means is that we can tell some things about the bird's history at a certain time scale with the bird's own DNA, but we can learn things about much more recent history by studying the parasite DNA because it evolves more quickly and accrues changes more quickly. And there are a whole bunch of reasons for that. One of them is that the DNA isn't as good at repairing itself when it copies. So these mutations accrue faster. And also the generation times of these parasites is much faster than the generation times of the birds. So that also allows these things to accrue more differences that allow us to look at really small time scales. So the parasites actually can tell us about population history of those condors. And that can be used to understand things about, sort of the conservation history and what's happened over time to these birds. So one of my undergrads is sequencing the genomes of these lice and sequencing the genomes of a bunch of other vulture lice, like turkey vultures and greater yellow-headed vultures and black vultures and king vultures, to try and understand the population histories of these things over evolutionary time. There's already been work done on the genomes of the birds themselves. So we're asking, what do the parasites tell us about that host population history? So it's kind of a cool use of these parasites, and one of many reasons that we probably don't want the parasites to go extinct either. As long as they're not killing the birds, they can be useful and interesting. And this is kind of the last endangered bird that I want to talk about, and then I'll talk about some little-known birds in the collection. This is a sharp-shinned hawk. And you have all, I'm sure, seen a sharp-shinned hawk if you've been out birding in the mid-Atlantic in the fall. I saw one today when I was out in my park. There were west winds and there were some raptors migrating. If you go to Cape May, the sky is just filled with these things on the right day, right? But this is a sharp-shinned hawk that's different than the ones that we see here. This is Puerto Rican sharp-shinned hawk. And it turns out that sharp-shinned hawk has a huge distribution. It's found from northern North America all the way down to southern South America. And it's in the mountains all over South America. It's in some lowland regions. And it's also on 3 Caribbean islands, Hispaniola, Puerto Rico, Hispaniola, and Cuba. And the Puerto Rican populations, we know, are highly endangered. So we were working with Peregrine Fund because they've been working on trying to hack birds. And they're also working on, so there's a fly called Philornis that lays its eggs on young birds. And those larvae from those flies kill those nestlings. So they can actually treat the nests to help defend those nests against this parasite that actually does kill the birds. So that's one of the many things that's knocking back the populations. Also, hurricanes are knocking back the population. So in 1997, there were 150 individual Puerto Rican sharp-shinned hawks. And as a birder, you could go and see them. There were some nests that were near roads and places where you could see them. Now it's pretty tough. You would really have to hike in. You have to get lucky to see one. I was in Puerto Rico. I saw almost every endemic, but I did not see this one. So in 2017, Hurricane Maria knocked back that population from 150 individuals to 19 individuals. So one of the most endangered birds on the planet, right? If it's a different species, it's even more endangered. It's even more important to worry about it. And what my lab's been doing is, so first of all, the populations have come up a bit. The work the Peregrine Fund is doing is helping those populations to increase. And one of my postdocs, Therese Katnack, the two of us worked with Peregrine Fund, and we started doing genetic work on all of these sharp-shinned hawks to try and understand, are the ones in North America, which are fairly abundant and common, different than the ones on these islands, different than the ones in Central America, different than the ones in South America. And it turns out there are many species of sharp-shinned hawks. If you look at that genetic data, these island forms are definitely different. All 3 of them are at least different enough from North America to be considered something different, but they're also different than each other. We know from the genetic data that birds on Hispaniola, Puerto Rico, and Cuba are not moving between those islands from their genetic data. So what that means is we need to actually pay attention to these different island populations and treat them as good units that need to be conserved separately. And these are actually modern specimens. So because we were working with Peregrine Fund, they had nestlings in captivity. Some nestlings didn't survive. Instead of throwing them in the garbage, they froze them and got them to us so that those are now specimens in the collection. There were only 3 specimens in collections, period, anywhere in the world. Now we have several nestlings, and we have 1 juvenile bird, the streaky bird with the wing specimen there. So at least we're able to, because we're working with this conservation organization, when they get their hands on material like this, it does get preserved for perpetuity, for, conservation work on the evolution of these things, all kinds of different things. Okay, let's just talk about a few new, little known and interesting things. Earlier on, we were talking about just chatting earlier, we were talking about the scarlet-banded barbet and discoveries in South America. A lot of these are discoveries in South America. And if you look back in time, in the early to mid 1900s, even 1950s, people pretty much thought that we were done describing new bird species. They thought that, insects, lots of new insects, new reptiles, amphibians, new fish, those things are out there, but birds are so well known that we're not going to find them anymore. And there was a famous ornithologist, Ernst Mayr, who was at the American Museum and then at Harvard University that actually wrote this in a publication. He said, we're virtually done discovering new bird species. Well, in 1963, a colleague of mine named John O'Neill, who was a student at the time and then eventually ended up at Louisiana State University, was on a trip to Peru, and an Agaruna indigenous person gave him some skins that they were using for decorations, ritual decorations. And so they weren't stuffed round skins, they were kind of these flat skins. But when he saw them right away, he knew this is something really strange. And it was this thing, this orange-throated tanager. And John and his advisor at the time published a paper on this specimen and described it as a new genus. It was named Wetmore thraupus, named after this guy named Alexander Wetmore, who is a well-known ornithologist from the Smithsonian. So basically, it was so different than any other known bird that they created a new genus to name it. And this orange-throated tanager is now seen by birders in parts of Ecuador, mostly in Peru, northern Peru. And it was sort of the first thing that sparked a lot of kind of adventurous expeditions to remote regions in South America that continue to discover new species of birds. So this is something that, when I was in graduate school, friends were going on trips discovering new species of birds. It's still happening, which is pretty cool to think that our planet still has unknown species of birds. This is another one of them. And John O'Neill was the discoverer of that bird. Well, this is a picture of John on a burro. And John discovered this one this was discovered by colleagues of John's in 1973. So it was collected by a guy named Reyes Rivera, and George Lowry and Dan Tolman, two researchers, named this in honor of John O'Neill. So it's called the Pardusco, and the species name O'Neillii is named after John O'Neill. So Nephelornis o'neillii. And this is a tanager. It's a weird looking tanager, not very colorful. And it lives in elfin forest at high altitudes in the Andes. And this was one of two different genera that were new genera discovered to science in the bird world between 1976 and 1980. So pretty cool, weird discoveries. And John used to have his email address was pardusco@aol.com. So he used that as his email address. This is another one of these Peruvian things, and this is a type specimen from our collection. And this was discovered by a guy named Mel Curator, who I mentioned collected specimens in Colombia, Peru, and Bolivia for the Academy of Natural Sciences. It's why we have one of the most important and most extensive collections of Andean birds in the world, because of the work that Mel Curator did. And Curator was in this inter-Andean valley in La Libertad, Peru, a pretty isolated area, in 1933, and discovered this toucanet. There still are no tissue samples of this toucanet in existence in any collection, really no modern specimens. There really are only specimens from this era. This is a cloud forest bird. It's if you've seen the greenish toucanets, like emerald toucanets, you kind of know on average they're in sort of higher elevation, cloudy, mossy kind of forest, and this one's like that too. So this is a pretty infrequently seen bird, and we have several in the collection because of Curator. This one is one of the modern discoveries from the Academy of Natural Sciences. So there is a modern era of discovery. Probably many of you knew of Bob Ridgely. Bob Ridgely was the Academy, and maybe you know Mark Robbins as well. Mark Robbins is collections manager at the Academy, and Mark is an eminent field ornithologist, knows neotropical birds incredibly well. And he was on a trip in the Ecuadorian Andes with Ridgely, maybe. Actually, this trip might have been not with Ridgely, because Ridgely's not on this paper. And basically, they discovered this cotinga that was different than another species that was found further to the north. And they noted that it was different, and they named this one after Van Remsen, who's an ornithologist at Louisiana State University, was curator of ornithology there. And this was described in 1994, and it's known from both Ecuador and Colombia. So it's considered vulnerable. It's very range restricted at high altitudes, and kind of a pretty cool discovery to find something new and flashy like that. Mark was also involved in the discovery of this thing called the El Oro parakeet. So this was Ridgely, and it was involved with this as well. And this is a parakeet that has a really small geographic distributional range. So you can see the map in the lower left there. That's southern Ecuador. And you can see there are just a few little purple dots. There are about 3 or 4 areas that have this El Oro parakeet. What's cool about this El Oro parakeet is this is a really big success story. So these guys discovered this bird, described it to science, and then Ridgely and colleagues started this thing called the Hokotoco Foundation. Some of you may have birded in Ecuador and gone to some of their reserves. Well, one of their reserves is home to these El Oro parakeets. So they've conserved a huge area of land that these El Oro parakeets nest in. They provide nest boxes for them to help increase population size. And the birds are doing pretty well because of that. One other thing that's kind of cool about this, it turns out that there was an unrecognized specimen of this thing in a collection in Tring. So the Rothschild collection in Tring in the UK had a specimen of this. A guy named Tom Schulenberg was visiting there and found it. And basically, this was discovered in 1980, but and was described in 1988. But this specimen had been sitting in a collection for a long time, unbeknownst to scientists. So sometimes there are cool little tidbits just hiding away in the collections as well. That's another way that people describe and discover new things. This is the namesake. This is where the name of the Hokotoco Foundation comes from, which has preserved all that land in Ecuador. And this is another one of these famous discoveries by Bob Ridgely and colleagues. Bob was involved in the discovery of this, but colleagues decided to name it after him. And it's poor form to name something after yourself. So these colleagues wrote the paper describing it. And Nate Rice was one of the colleagues on this. Nate sequenced DNA from these for his dissertation and did the molecular analysis of all of the birds in this group to assess how different this was than those things. And it's strikingly different. It looks nothing like any other ant pitta out there. It sounds different. That's how Ridgely found it. He heard an ant pitta that he knew was different. This is often how we find things in the neotropics. Sound is really important, just like it is here. And they collected a handful of specimens a few years later. And what's interesting is they've also conserved a big area for this bird in Ecuador. But what's cool is John O'Neill led an expedition many years ago when I was a graduate student at LSU to northern Peru, to some regions just across the border from Ecuador. And they were up in high elevations where there was habitat as far as the eye could see, and at this particular elevation. And they found Hokotoco ant pittas there in Peru as well. So Hokotoco ant pitta now has a much larger distribution than we once thought it had, which is great news. And then this one is a bird called the white-winged potu. And the white-winged potu was initially only known from two 19th century museum specimens. And I don't know if any of you know Rob Beauregard. He lives near me in Wynwood. Rob is an ornithologist who's from the Philadelphia area, went to graduate school at Penn, studied raptors, and then went to Manaus and worked in Manaus for many years. And he worked with a friend of mine named Mario Kohnhoft, who's also from the US. He's from Massachusetts. And Mario spent a bunch of time at this famous forest fragment project called the Manaus Forest Fragment Project, where they study the effects of forest fragmentation on bird populations. And Mario heard this bird singing and could not figure out what it was. And he kept on telling his local field hands, hey, what is this thing? I recorded it. Do you have any idea what it is? And they would tell him that it was the Curupira, which is like a mythical creature, all kinds of funny stories like that. And, he was just convinced that it was something interesting. Well, eventually he went up on a rope. He pulled himself up into the subcanopy on a rope and played recordings and finally saw this bird, and it was the white-winged potu. And potus are related to our nightjars. They sit upright. They kind of they're very Muppet-like. They have big eyes, and they basically, they forage by flying up and grabbing moths and things on the wing, but you'll see them sitting on fence posts like common potus if you go to open areas in South America. But this thing lives in the subcanopy of rainforest, was really unknown. And the interesting thing is, of these couple of specimens that were known, they were from eastern Brazil, northeastern Brazil. And Mario was smack dab in the middle of the Amazon when he heard this thing and found it. And they were able to collect some specimens. Rob Beauregard prepared some of them when they described the rediscovery of this bird. And now it turns out it's found all over the place. It's found in lots of sandy soil forest patches. It's found in the Guianan Shield. That's where this specimen comes from Guyana. It's found in so many areas that, it's hard to believe that it was unknown in life. And not only that, but the birds in eastern Brazil in the Atlantic forest have been refound. So because of Mario's rediscovery, we now know what this, what this bird is like, what it sounds like. And it basically has like a downward whistle, like a, you would think that something like that would be something you'd notice, but apparently for maybe centuries, it was hidden away. Um, this one's just one of my favorites because of the name. I mean, who would not want to see an inaccessible island rail, right? If you could pick a bird that you wanted to see. And this is found on Inaccessible Island, which is out in the middle of the Atlantic. So this is a map that's Southern Africa, and you can see that little speck is Inaccessible Island. These are considered to be the smallest, one of the smallest flightless birds in the world. These are a flightless rail. And this island group is in the Tristan da Cunha group, if you've heard of that. Not a place that very many people go to. And they're supposed to be fairly abundant. But of course, they're vulnerable because they're flightless and they're on the ground. And they're found in sort of areas of kind of like bunch grass. And if you look, at least the last time I looked on Zeno Canto or the Macaulay Library, there are no recordings of the sound of this thing. And there are only 2 eBird observations the last time I looked. So this is a bird that not many people have seen, and we have some specimens in the collection. Anyone here seen it? Yeah, probably not. This one is kind of cool, not because it's rare. This is broadwing hawk. This is a cool story because this is the type specimen of the broadwing hawk. Anyone know where this specimen comes from? I don't think I gave it away on the slide. It comes from right here in Philadelphia. Alexander Wilson, when he was working on studying birds of North America and was writing his book, went to Bartram's garden and collected this specimen in Bartram's garden. So the type specimen, the icon, the thing that we use to define what broadwing hawk is, came from Bartram's garden, which I think is pretty crazy. And we still have the specimen. It's extant. People can measure it and study it. The other cool thing about broadwing hawks is we know that this vast body of them nests all across northern North America. We have them nesting here in Pennsylvania. They nest up all the way into Canada. It's a summertime bird of, the Poconos and, all the way up in, the northern US. And they migrate in crazy numbers, right? You can see huge kettles of them migrating through, and those go down to the Andes of South America mostly, although they're in other parts of South America as well. Well, what's cool is if you go to the Caribbean islands, there are populations on many Caribbean islands, including Puerto Rico. And it's a very similar story to the sharp-shinned hawk, except we don't know the end of the story. The beginning of the story is that there are resident populations that do not migrate on those islands. And the question is, are they genetically different on all those islands? So this is one of these projects that we're starting now to look at these things and try and understand. We're working on this with Peregrine Fund as well. Are these island populations of broadwing hawks different than the mainland populations that are so abundant? Because these island populations, they're not doing as poorly as sharp-shinned hawk, but some of them are having trouble too. How many of you have seen a corncrake here in the US? Oh, yeah. So most people have seen them in the old world. It used to be that you could see them occasionally in North America. There are around 17 records from just here in Eastern North America. And it seems like they sort of petered out. They stopped showing up as vagrants in North America. And then a few years ago, I was actually on Facebook and I saw someone reporting a dead corncrake here in Pennsylvania. And of course, I was already here at the Academy. So I said, oh, we want that thing. Like we don't want it to get thrown in the garbage, And so through Facebook and friends of mine, I got in contact with the people, and Nate Rice hopped in his car and drove up to Wayne County, Pennsylvania. And this bird right here with the wing spread, this is a very nice specimen prepared by Nate Rice. One of the nice things about nice specimens like this is they will last hundreds and hundreds of years because they're put together really well. And there's a lot of data with that specimen. Unfortunately, that was killed by a cat. So another one of these examples of a cat-killed bird. What's crazy is the next year, so I should mention that, of course, I wasn't the only museum curator saying we wanted this specimen. There were other regional, the American Museum was saying they wanted it. Everyone was saying they wanted it. But of course, we always, if there's a dead bird in New York, I don't fight the American Museum for it. If it's in Pennsylvania, they don't fight me for it. We're very amicable. So they said, oh, because we all care about just it's in a collection where people will have access to it for perpetuity. That's the most important thing. So they said, you can have that. Well, a year later, a corn crake was found in New York. And it was found right along a roadside, which of course, all I'm thinking is a car is going to take this thing out eventually. People were seeing it along the roadside. And Glenn Seaholzer, who's a Pennsylvanian who went to Cornell and actually works at Cornell now and was at the American Museum doing a postdoc. Glenn went out to go see it. And I think right when they pulled up, it was hit by a car. And now that's at the American Museum. So they got their corn crake. Their version. So we actually have 4 specimens from North America. We have several from New Jersey, one from Cape May, one from Salem, and one from Burlington, New Jersey. So they were something that did show up more frequently on the coast in the past. This one is a cool story because this is a screech owl. We all know screech owls, right? We all know megascop screech owls, like our eastern screech owls, but this is the South American equivalent. This is called tawny-bellied screech owl, found all over the Amazon basin. And then there's a close relative called the black-capped screech owl that's found in the Atlantic forest. And we did a study of these things because we wanted to ask that same question. Are all these populations the same or different? There are all these different vocal types. And the Amazon, any of you that have been to the Amazon, when you're flying over in a plane, it just looks like a giant green carpet. But there are these major rivers, and these major rivers basically form barriers. A lot of birds will not cross major rivers. Even a screech owl like this, it's an understory bird, will not cross over a major river. Little songbirds will not cross over major rivers. So a lot of the species that you find in South American forest are really endemic to these little small local, we call them interfluves, these regions between major rivers. So there's a lot of endemism in Amazonia. And if you want to see all the birds in Amazonia, you have to actually go to a lot of places. You can't just go to Peru or Ecuador. You need to go all over Brazil. You need to go to the Guianan Shield. You need to visit all these major areas. Well, it turns out that these tawny-bellied screech owls match that story, that all these different populations from different places were genetically highly different. Their songs were very different in these different places. But if you looked at their plumage, you can't tell them apart. They're all what we call polymorphic. So like our screech owls, there are rufous morphs and gray morphs. These things have all these different color morphs. There's a lot of variation within populations. And because of that, when you look at specimens side by side, you cannot tell them apart. Well, there were already some taxonomic names out there in the literature. So there's this thing, Megascops watsoni, that was published in the literature. But the problem was we didn't know what that name applied to, which one of these populations around the Amazon basin did that name apply to, because the specimens are often old. They don't have specific localities that were originally used to describe these things. So we really needed to know what they were genetically related to. So we did this by sequencing DNA from these old type specimens in our collection. We sequenced DNA from them. We found out which population the name applied to. And what that allowed us to do was we basically found out that there were a couple of unnamed populations. There were 2 things that were unnamed, and we were able to name those. One is called the Alagoas screech owl now, and the other is called the Xingu screech owl. And these are both found in areas that are fairly endangered. So it was very important that we were able to characterize them, because now people can, they're on the endangered list in Brazil now, for instance. So those areas can be protected. And this is just a map showing those 2 species, the Xingu screech owl and the Alagoas screech owl. And you can see that the Xingu screech owl is that kind of lime green, kind of rectangle in the middle of the Amazon, sort of just south of the mouth of the Amazon. And then the Alagoas screech owl is that green spot at the bump of Brazil. So both of these have fairly, limited distribution. And that green splotch, that whole eastern part of the Amazon south of the river, is very damaged part of if you fly over it now, it's a patchwork of forest. There are still some big patches, but it's a much more endangered part of Amazonia than Western Amazonia in like Peru and Ecuador is. So I think I only have, let me just check, 2 more or a couple more little things here. These are just some weird things in the collection. This is a hybrid Junco white-throated sparrow. And believe it or not, these are more common than you would think. There are eBird records of these. You can look them up on eBird. You can actually listen on eBird and see what they sound like. There's one recording on eBird. What do you think it sounds like? It actually sings like a junco. It's a trill. At least the one that's on eBird is a trill like a junco. But there are 128 observations of this hybrid combination in eBird, and 43 of those have photos. So we happen to have a specimen of this thing, but pretty cool, weird bird. One of the other weird oddities that we have is this one, and this is actually the last thing that I have to show you. This is what's called a bilateral gynandromorph, and this is an orchard oriole. And bilateral gynandromorphs are literally half male and half female. So one side of the body has testes, one side of the body has ovaries, and the side of the body that has the testes also has the coloration of a male, and the side of the body that has the ovaries has the coloration of a female. And this is pretty unusual. I actually have not heard of anyone finding any of these in orioles before, but for some reason in cardinalids, birds that are related to cardinals, it seems to be a more common event. So northern cardinals, it's fairly regular, and the bird on the lower right-hand corner is a bilateral gynandromorph northern cardinal, and you could see, it's right down the middle, male, female. Then there's a rose-breasted grosbeak that's male, female. It's an immature bird, so it doesn't show the full coloration of the male on the left side, but you can see it's pink armpit and kind of a yellowy armpit. And then evening grosbeaks are also known for this. And these evening grosbeaks are, the one in the middle is from the University of Minnesota's Bell Museum collection. And you can see, that cut right down the middle, half male and half female. So, thanks for your attention. There's so many other cool stories and interesting specimens in the collection, and this is just sort of a smattering of them. Thanks for your attention.